All right, so good afternoon, everyone. It's my great pleasure to introduce myself and our project in front of you today. We come from very different worlds, so I will try to be as clear as possible on the topics we study. And I'd like to start by showing this tree of life that was made by a PhD student from the Pasteur Institute. I really like this tree because it beautifully shows that microorganisms make up most of living diversity. So, we humans are over here on the tree, and as a reminder, the clay is so big, opisthocomta, that it contains both fungi and all animals as well. So it's a good reminder that we humans are completely insignificant in the scales of the tree of life. And so despite this huge diversity, there's something that is common to virtually all forms of life, is that we are all infected with viruses. Not only us humans, but also plants, archaea, and also bacteria. And so these viruses can take lots of different shapes, particularly like these champagne bottle shaped viruses that infect archaea. Wait, let me just take a pointer. Yes. But also these lemon shapes are pretty nice as well. And so basically the arms race between viruses and host cells is extremely ancient, estimated to be as old as life itself. So viruses have, however, been a huge selective pressures on their host. And so the viruses that infect bacteria are called bacteriophages, or phage eaters, or just phages for short. And as in any other kinds of viruses, they take lots of different kinds of morphologies. They can be tailed or non-tailed. They can carry also DNA genomes or RNA genomes. It can be double-stranded or RNA-stranded. So there's a huge diversity of the phages, and there's also a diversity of lifestyles. So typically, the most studied phages that make up most of model phages that have been studied for decades are virulent phages. These virulent phages, basically, when they encounter the host, they first absorb to the bacterial cell, then they inject their DNA content into this bacterial cell. This DNA content starts expressing the first early genes, and this includes genes that allow phages to take over the host, typically by blocking host processes. And so at this stage, the bacterial cell becomes basically a factory for the production of viral particles. And so intracellularly, these proteins from the viral proteins are assembled into new variants. And finally, the phage would lyse the cell. So this is actually an active process. Phages produce specific proteins to lyse the cells at a dedicated time. And so you can expect typically that a single infection event will lead to the release of something like 100 new variants into the environment. But on top of this, some phages are not only virulent, they are called temperate phages, which means that during infection, they can either go through the lytic cycle, or they can stochastically integrate into the bacterial chromosome and enter a dormant state that is called lysogeny. And what's interesting is that within these dormant states, the fitness and the survival of the phage is tied to the survival of the host. And so typically the evolutionary pressures that drive the selection of these phages are pretty different between virulent and temperate phages. And finally, there's another type of phages that are called filamentous phages that also are pretty different, because they can continuously release phage particles from the host, but without lysing the host cell. So I'm not going to go too much into detail on these phages, because we have mostly focused on virulent and temperate phages. And so phages have a major impact on pretty much any environment. They're considered to be the most abundant biological particles on our planet, being at least as abundant as bacteria on Earth. If you take a milliliter of seawater, you'll find 100 million phages in it. It goes up to 1 billion phages if you take a gram of soil. And so to highlight the importance, it basically lies something like almost a third of marine bacteria every day. So they also participate in a huge turnover of organic matter, pretty much in any environment. Okay, so phages are everywhere. They absolutely have major pressures on their bacterial hosts. So why do bacteria still exist after all? Well, bacteria still exist because, as you guessed, they have an immune system, and so bacteria can defend against these viruses. And so you'll see through this presentation that the field of bacterial immunity against those phages is both very ancient field in terms of biological ties, but also a very new field of research. And so we'll start with the early days of bacterial immunity. It all started actually in the 50s when people were studying phages as molecular tools to decipher the principles of life, and especially these were very useful tools for genetics. And so there was a famous experiment that involved phage lambda, which is a very classical model. And this phage lambda was first passage on two different E. coli strains that are called S and C. And what people observed is that the infectivity of the phage depends on the host on which it was passaged. So typically, if you take this passage phage and you plaque it on a strain of E. coli S, you'll see that the phage that was first passage on E. coli S has a pretty high fitness, can infect. So this is a plaque assay, which measures the number of plaques in a phage sample. So these plaques are formed by the lysis of the bacteria. So one plaque is equivalent to one initial phage that touched the agar. But when the phage was first passage on E. coli C, the infectivity was much decreased. And so that was a very intriguing phenomenon that was at the time called host-induced variation, meaning that the infectivity depends on the last host on which it was passaged. And actually, this was the first manifestation of the most widespread bacterial defense system, which is called restriction modification. The general principle of restriction modification, or just RM system for short, is that you have two enzymes. You have first a methylase, and this methylase will modify a defined position, defined motif in the host genome. And then the second component is the restriction of the nuclease. This nuclease will cut any DNA that does not have the modification. So this is a simple way to discriminate self versus non-self. So this methylation and this restriction are specific. For example, the EcoR1 enzyme, which is widely used in laboratories for restriction cloning, recognizes the specific motif GA8, TTC, etc. And actually, this defense system is always the beginning of an arms race, because of course, if phages exist today, it means that they found ways to inhibit those defense systems. And so typically it was found early on that some phages evade the RM system by acquiring DNA modifications in their genomes. And a famous example is that of phage T4, which is another very classical phage model, in which all the cytosines in the genome are replaced by a glycosyl modification. And this completely changes the DNA properties, and the restriction enzyme cannot recognize anymore the phage DNA. But then in turn, this puts a pressure on the bacterial hosts to evade and find a new defense strategy. And for instance, another kind of RM systems are type 4 restriction modification systems. And actually, these restriction enzymes, they function the other way around. They specifically recognize modified DNA. And so actually the fight keeps going on for a while because phages then found ways to evolve proteins that are called anti-restriction proteins that can block specifically some kinds of restriction systems. And so this was the early days of restriction modification, because these were discovered more than like 60 years ago. But there's another very famous family of defense systems that you all heard about that was discovered slightly more recently, and this is CRISPR-Cas systems. So CRISPR-Cas systems were first identified actually a long time ago in the 80s, where scientists identified repeated regions in the E. coli genome, but then in other kinds of genomes as well, in prokaryotes. But the breakthrough study came in 2008 when scientists found that these repeated regions are typically found in what is called a CRISPR array. The content of these repeated region matches the resistance profile to phages. So typically they found that if you take an initial strain of Streptococcus that carries two of these spacers, it is sensitive to a phage that is called phi-858. But if you evolve this bacterial host to acquire resistance against this phage, then the CRISPR array acquired two more spacers, and this would be sufficient to make the bacterial host resistant to the phage. And so it was the first time that there was strong evidence that CRISPR-Cas was actually an immune system against phages. And another breakthrough study was published in 2012 showing that these CRISPR arrays are used by the Cas9 nuclease serves as guides to recognize the foreign nucleic acid and to cleave it in a sequence-specific manner. And so basically the general principles of CRISPR-Cas systems is that upon the first infection, there are pieces of the invading DNA that can be integrated into this CRISPR array, which forms a memory of past infections. And then on later infections, this CRISPR array can be transcribed, processed into pre-CRISPR RNAs, and these CRISPR RNAs can guide the Cas9 nuclease to specifically cleave and eliminate the invading nucleic acids. And so you all know about the Cas9 nuclease, but actually it's only a small part of the huge diversity of CRISPR systems. Many types and subtypes of CRISPR systems exist. Typically there are two classes. In the class 1s you have multiple enzymes that multiple protein components that together form this interference complex, whereas in class 2 CRISPR-Cas systems you have a single enzyme. And there are very interesting subclasses. For instance, there are some the type 6 CRISPR-Cas systems. They don't target DNA, they target RNA, etc. So Cas9 has been receiving a lot of attention, but there are also lots of cool technologies that derive from other kinds of CRISPR systems. All right, so if we are back like 10 years ago, we know that bacteria evolved restriction modification systems to defend against phages, and with these restriction modification systems are the most abundant defense systems so far, found in like 80% of bacterial genomes. Then we also know CRISPR-Cas systems found in around 40% of bacterial genomes. And of course, both of these families of defense systems led to major breakthroughs in gene editing and molecular biology. And in parallel of these discoveries, people were also studying the innate immune system of other species across the tree of life. But what's quite intriguing at this stage is that RM systems and CRISPR-Cas are absent in virtually all known eukaryotes. There's not a single eukaryotic genome that contains a restriction enzyme or CRISPR-Cas system. And in parallel, very elaborate strategies for defense were discovered, especially in animals and in plants. And this includes inflammasomes, tear signaling, C-gasting, and many more. And altogether, this led to a paradigm which was that, okay, prokaryotes and eukaryotes have nothing in common in terms of immunity, simply because the enzymes, the actors that we know are not found in the other domain of life. And this could be explained simply because the viruses that infect those organisms are just super different. So there's no reason why they would have similar defense systems. But in the rest of the talk, you see that this was an old paradigm that was shown to be wrong. And it all restarted something like 10 years ago. And the way it restarted was when Kira Makarova, who's a bioinformatician at the NIH, made a very peculiar observation. So these are bacterial genomes. So you see that so each arrow is one gene, and so these genes are typically organized in operons that gather similar functions in prokaryotes. And so she observed that genes that are called CRISPR-Cas systems or that are called RM systems would be found next to one another in bacterial genomes. And so this led to the hypothesis of defense islands, which would be specific regions of bacterial chromosomes that would be dedicated for the integration and the accumulation of multiple defense systems. But then, considering this hypothesis, if you find that operons of genes are very frequently found within those defense islands, then it's a good sign that maybe those operons would form actually defense systems that are yet to be discovered. And this is the approach that was employed by the lab of Temsorek in 2018, that systematically investigated the location of all microbial genes next to known defense systems. And this approach was very successful because it led to the discovery of 9 new defense systems in a single study. And so the predictions were purely bioinformatic, but then this was combined with experimental work. You can predict defense systems using this association next to known defense systems. Then you can synthesize the DNA encoding these candidates, introduce it into a model like E. coli or Bacillus subtilis, and then infect it with phages. And ideally, you should see that the strain with the defense system should be protected against phage infection. And so this was 9 defense systems in 2018, and this approach was expanded then in 2020, 2022, using a very similar approach. And altogether, this led to the discovery of over 50 new defense systems in just a few years. And so beyond defense islands, it seems that another source of defense systems against phages and against mobile genetic elements in general seems to be mobile genetic elements themselves, and especially phages. So typically I mentioned initially that some phages are called temperate phages. They can, during infection, integrate their DNA within the bacterial chromosome. And when they enter this state called lysogeny, they're basically dormant, and their survival is tied to the survival of the bacterial host. And so it's frequent that these phages actually carry genes that are not essential for their own cycle, but provide a fitness benefit to the bacterium to indirectly enhance their own fitness during lysogeny. And so typically there's a famous phage that is called phage P2. It typically infects enterobacteria, especially E. coli. And these phages have lots of essential genes, like structural genes, replication genes that are shown in blue. But it was known for some time that it also carries 2 genes that are not essential for phage infection. And actually, P2 phages are very widespread. Basically, like 70% at least of E. coli strains have at least one P2-like phages integrated into their chromosome. And if you look at these other kinds of P2-like phages, you see that the phages are very conserved, and then there's a huge variable locus located within what we call the defense hotspot. And we could show that actually this defense hotspot is a great source of defense systems, with like hundreds of potential defense systems integrated in this specific spot. And so the idea is that P2-like phages can integrate within the host chromosome during an enterolysogeny, and when they do so, they equip the bacterial host with a defense system that will protect the cell against competing viruses. And so this is a great example of a competition between viruses. And so far, actually, defense systems have been discovered not only on these temperate phages, but in virtually any kinds of mobile genetic elements that can use bacterial cells as a transient host. Okay, so just in just a few years, we started from our knowledge of restriction modification, CRISPR-Cas, to basically a huge diversity of defense systems. Today, around 300 defense systems have been discovered, and over 90%, maybe 95% of them were discovered since 2018. So it's been a major revolution for microbiology, with lots of very new insights for the field, or even of immunology in general. And for example, this led to multiple new kinds of molecular mechanisms. And I just want to highlight 2 examples. So typically, these defense systems can be single proteins or multiple multi-gene systems. And there's an example that is called DSR. And this DSR, it's a single protein that would directly sense protein from the structural protein from the phage. And so when it senses this protein, it unleashes an effector domain that degrades the essential metabolite NAD+. And so this shuts down the cellular metabolism, and this blocks infection. Another example is a defense system called CAPREL, which once again is a single protein in which there's an antitoxin domain that blocks the toxin domain. And so during infection, there's a capsid protein that can be directly sensed by the antitoxin domain, and this releases the toxin domain. And this toxin functions by inhibiting translation through pyrophosphorylation of tRNAs. So this is just two examples, but we keep seeing new papers every week virtually that showcase new kinds of molecular mechanisms that were never described before. Okay, so how does this relate to the abundance of CRISPR-Cas and restriction modification systems? Well, at this stage, RM and CRISPR-Cas are still the most abundant defense systems found throughout prokaryotes. And then there's a huge diversity of rare systems. So the bottom line is that very few systems are abundant, and lots of systems are actually pretty rare. So we are very far from reaching saturation. We're very far from having discovered all defense systems. And this defense system can actually accumulate within the same genome, because they're found in defense islands, they're found in prophages, in plasmids, etc. And so typically, if you the average genome would encode something between 5 and 6 defense systems. Actually, these figures are a bit outdated now. It's more like 7 to 8 defense systems that can be found in the typical genomes. And I think the champion so far is the cyanobacterium that can encode like 48 defense systems or something like this. So these systems can accumulate and be lost also at very high rates. And the consequence of this is that if you take 2 strains of the same species, well, they will encode very different kinds of defense systems. Here's an example with like E. coli strains that can encode. So some of these defense systems are pretty conserved across E. coli strains, like RNs, and to some extent CRISPR-Cas systems. But then the distribution of the systems is very patchy, and this is once again due to the fact that most of these systems are encoded in mobile genetic elements, so they can be acquired and lost very quickly. And so another aspect that was underestimated before is the importance of regulated cell death and cell dormancy in bacterial immunity. So for a long time, bacteria were considered as unicellular, like planktonic cells that do not have an interest in having don't have any interest in being altruistic in their defense mechanisms. But actually, natural microbial communities are organized in colonies of isogenic cells, so that typically, if you have a single infection event, this infection event will lead to the release of multiple phages, which will eventually lead to population collapse. So it's actually beneficial that some defense systems employ regulated cell death or dormancy to block the growth of the infected cell. And this would block this would block the release of phages that would otherwise wipe out the entire cell population. And actually, we realized that these defense systems are super abundant. They're found in the vast majority of sequence prokaryotes. And we believe that in terms of evolution, they're so successful because they are very modular at the genetic level. So typically, these defense systems encode a sensor that would detect a specific signal during infection. And this sensor would then transmit the information to the effector, which would execute cell death. And then when you look at microbial genomes, you realize that there's a defined set of protein domains that can be effectors that trigger cell death, for instance, through DNA degradation by degrading any nucleic acid present in the cell, or by degrading the essential metabolite in AD+. And then conversely, there's also a defined sets of protein modules that act as sensors. And then there's a huge rate of recombination between these effectors and these sensors. Typically, a given family of defense systems can accommodate multiple types of effectors, and conversely, a type of effector can be found across very different families of defense systems. So these kind of behave like Lego bricks. They can be arranged and recombined, and this the result of this is that this completely drastically increases the molecular diversity of these defense systems, starting from a very, very limited set of building blocks. So we behave that there's something constitutional in the nature of these defense systems that makes them so successful in terms of evolution. And so there's a significant perspective that that was also emerging in the past few years, is that among those protein modules, may they be effectors or sensors, we're starting to realize that some are actually common between prokaryotes and eukaryotes, and it seems that some are conserved across domains of life. And it all started with a pathway that is called cGASTing. cGASTing is an immune pathway that we have in our own cells. And in our own cells, we have an enzyme that is called cGAS that senses nucleic acids in the cytoplasm. And this is typically a sign of infection because DNA should be only in the nucleus. And when cGAS senses a double-stranded DNA, it starts producing a molecule that is called cGAMP, cyclic GMP-AMP. And this signaling molecules binds to the STING receptor to trigger the interferon response. And so this will alert the organism that an infection is going on. And actually, cGASTing was discovered in 2013. And based on this defense system that we have in humans, a defense system called cBAS was later discovered in 2019. And cBAS encompasses very similar components because there are cGAS-like enzymes in bacteria that can similarly produce signaling molecules. And these signaling molecules can bind to a receptor domain. But this time, this receptor domain is directly fused to an effector domain that will trigger cell death. And so this was the first evidence that an immune pathway could be actually conserved between prokaryotes and eukaryotes. And if you look at the phylogeny of cGAS or the phylogeny of STING, it becomes very clear that cGAS-STING was actually invented by bacteria as a defense mechanism against phages and was later on transmitted to other organisms over the course of evolution. And actually, this was just the first example, because in the past few years, we have seen the multiplication of these kinds of cases. For instance, humans have an enzyme that is called SAMHG1, which is actually a restriction factor against HIV. And this enzyme during a viral infection can deplete all dNTPs, which are used as building blocks for viral replication. And so this blocks infection. Actually, similar enzymes exist in prokaryotes, and they degrade the nucleotide GTP to block similarly viral DNA replication. Another example is that of viperins, which are enzymes also found in humans that take the nucleotide CTP and remove one of the OH groups to make a chain terminator. So when it's incorporating into a nascent RNA chain, it blocks completely the incorporation of the next nucleotide. So, and similar enzymes also exist in prokaryotes that defend against phages. And sometimes much more complex proteins are also conserved. For instance, NLRs are typical immune proteins that are found across eukaryotes. They encompass repeats on the C terminus that bind to a specific invader component like a protein. And this triggers the leucomerization of the protein into what is called a resistosome in plants or an inflammasome in animals. And this leads to the activation of the effector. And actually, very similar proteins exist in prokaryotes. And similarly lead to the activation of an effector domain, which is responsible for triggering cell death. And so importantly here, most of this knowledge that we have acquired over the years on this conservation was permitted thanks to our prior knowledge on eukaryotic immunity. We already knew SAMHG1, viperins, NLRs in eukaryotes, and this was used as a basis for the discovery of new defense systems. So in a way, the field of bacterial immunity greatly benefited from what we already knew in eukaryotic immunity. And so typically, we also observe that some defense systems can accumulate several components that are also conserved in eukaryotic immunity. Typically, there's one of my previous projects started when we observed an operon of two genes that encode a TR protein and a caspase-like protease, and these were typically found next to known defense systems in microbial genomes, which was a good hint that these would be a defense system. And actually, we could validate this hypothesis by showing that if you take an operon from Pseudomonas and you put it into E. coli cells, it will protect E. coli cells against phage infection by phage T6. But if you mutate these two, either of these components, you lose the defensive phenotype. So this shows that this operon is indeed a new defense system. And we are very excited about this defense system because both tier and caspase-like proteases are well-known players of innate immunity. If we start with tier domains on the left, tier domains are well-known players of animal immunity. Tier stands for TLR and interleukin-1 receptor. And so typically these function as protein-protein interaction domains during infections that lead to the recruitment of adaptor proteins to lead to start a signal transduction. But tier domains are also enzymes in plants and in prokaryotes. And these enzymes degrade the essential metabolite NAD+, and they can do it very quickly, in which case they function as effectors that deplete the NAD+ in the cell to block infection. But they can also do it very slowly, in which case they function as producers of signaling molecules to activate the downstream effector. And on the other hand, caspase-like proteases are super well-known proteases in animal immunity. They are involved in cell death as well. And typically we have initiators and executioners without going into too much details. These participate in inflammation and apoptosis in humans. And so we managed actually to solve the molecular mechanism of this defense system by showing that during infection, the tier protein senses a phage component that we actually don't know yet. And upon sensing, it starts producing a new kind of signaling molecule that we call N7-CA ADPA. And so this is produced from the essential metabolite in NAD+. And once this signaling molecule is produced, it can bind to a protease that will then be unleashed into a promiscuous protease that will degrade multiple proteins in the cell. And the effect of this is that this massive protein degradation blocks any further replication of the phage in a manner that is similar to some caspases that we have in our own cells. And so this showed that caspases are one more component of programmed cell death and innate immunity that is conserved across domains of life. And it also shows that, one more time, that these tier domains in bacteria can produce very diverse types of molecules. And actually, this is very insightful for the field of innate immunity in plants, because tier domains in plants were also shown to produce a range of signaling molecules in immunity. And actually, we are starting to see that plants and bacteria produce partially overlapping sets of signaling molecules. All right, so if we step back a little bit, we now know around 10, 12 protein domains, so in most cases these are protein domains, sometimes it's the full proteins that are actually conserved across domains of life and participate in innate immunity. So when I say conserved, they are not conserved in all organisms, but they are found in very diverse taxa. So typically, cGAS-like enzymes are found in some bacteria, like 15% of bacteria. They're also found in some archaea, and they're found in most animals as well, but they're not found in all eukaryotes. And actually, if you look at the phylogeny of these cGAS-like enzymes, you can observe this kind of phenomenon in which so all bacterial enzymes are shown in gray. And you can see that in a few instances over the course of evolution, so eukaryotic sequences in pink emerged from bacterial sequences. And so this is a good sign that eukaryotic organisms acquired cGAS-like enzymes several times over the course of evolution. And that this is a good hint that this occurred through horizontal gene transfer at some point. And importantly, this can also lead to a very insightful hypothesis, because cGAS, which is this well-known human enzyme, is found in this red clade of CGLR superfamily. But this evolutionary analysis also led to the identification of another clade of eukaryotic enzymes that also seem to emerge from bacterial immune enzymes. And so this is tempting to speculate that these distinct family of enzymes also have immune functions in those organisms. And so, as I said, so far our knowledge of eukaryotic immunity led to the discovery of new bacterial defense systems. But so we're starting to realize that we've discovered multiple new defense systems against phages in prokaryotes. So why not try to actually do the opposite? Can we now discover new players of eukaryotic immunity based on what we discovered in bacteria? And so typically I was mentioning this modularity of defense systems. So typically in CBAS defense system, you would have the sensor, which is producing signal molecules, and one of multiple possible effectors. This can be a nuclease, or this can be a phospholipase. But sometimes you look at other microbial genomes and you find a protein domain of a non-function. But based on this association, you can make the assumption that this would be a new type of effector that you don't know yet about. Actually, this observation led us to discover a new type of effector that we name ATP nucleosidases. So it's going to get kind of molecular, but basically we found that if you incubate purified enzymes with a mix of 4 nucleotides, it's able to specifically degrade ATP molecules into adenine and ribose triphosphate. So this is basically the reaction that it's doing. It's chopping off the head of ATP, and so ATP is being such a central molecule in pretty much any kind of energetic process. The effect of this reaction is to deplete the pool of energy in infected cells to block viral infection. And so this was a new effector that we found in bacterial defenses, but by performing deep homology searches in eukaryotic genomes, we realized that very similar protein domains exist in multiple organisms. For instance, they are found in a lot of fungi where they can associate with typical immune domains like stings that senses signaling molecules, or in those as the effector of those proteins that can form inflammasomes. Beyond fungi, we also found them in sponges, in corals, but also in pretty much any protostomia, so including insects, including Drosophila, C. elegans, where it associates with the domains that it was shown to be a DNA sensor. And so altogether, this we are actually able to show that these eukaryotic ATP nucleosidases have the same molecular activity as the bacterial ones, and this combined with the typical architecture of immune proteins strongly suggests that ATP nucleosidases are widespread immune effectors in eukaryotes as well. And expanding this, recent beautiful work from the lab of Aude Bernheim and Enzo Poirier showed that other components of bacterial immunity can be found throughout the tree of life. And an example is that of the sirtuin domain that I showed previously. So this sirtuin domain in the DSR defense system is responsible for depleting the essential molecule innate. Actually, this enzyme has structural homologs, including in the human genome. And this was a very big discovery because it suggested that this human protein that was named CIRAL might have an immune function in eukaryotes as well. And so when they analyzed the genetic environment of this gene, they realized that it was frequently found across very distant organisms, frequently found next to a gene that is called TRAP. And TRAP is a well-known gene that participates into a pathway that is called the TLR pathway. TLRs are immune receptors that are located on the cell surface of our own cells, and upon stimulation by a pathogen-specific ligand, they recruit the TRAP protein and other adapter proteins, and this leads to the activation of diverse pathways that will mediate an immune response. And so they did this critical experiment in which they took cells that have or do not have the CIRAL proteins, and they measured the cytokine release upon stimulation, and they found that basically if you remove the CIRAL gene, there's no cytokine production anymore. They could also show that cells that do not have CIRAL are much more sensitive to replication of Salmonella enterica, showing that the CIRAL gene is indeed a new immune gene. And this is a beautiful illustration of what we can learn on our own immune system, starting from bacterial defenses. So to wrap up, I hope I convinced you that bacterial immunity is far more complex than previously envisioned. We are realizing now that bacteria have a huge arsenal of defense systems beyond classical RM and CRISPR-Cas systems. And we're also realizing that innate immunity across domains of life is actually connected through evolution. And in this way, bacterial immunity can help us to discover new immune mechanisms in eukaryotes using comparative immunology. And going further, we can hypothesize that if bacterial viruses and eukaryotic viruses encounter similar defenses in their hosts, it's tempting to speculate that maybe they also evolved similar ways to evade these immune pathways. So we anticipate in the future, in the next few years, that we'll start discovering immune evasion principles that are also common between phages and eukaryotic viruses. So with this, I'd like to thank my wonderful team. So I started my lab almost 2 years ago, and it's been a wonderful adventure so far. I also want to thank our funders and you for listening.